With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Question here says, in biolistic or gene gun method, DNA is coated with what? First of all, what is this biolistic or gene gun method? These are the method which are used for transformation of cells. What is transformation of cells? First of all, we need to understand, for example, this is our vector plasmid DNA and we inserted a gene of interest here. Inserted a gene of interest here. This is our gene of interest. This gene of interest has been inserted in our vector DNA, vector plasmid DNA. This is a circular DNA which is an extra chromosomal DNA and it multiplies irrespective of the main host cell's DNA. So this is our vector DNA. Vectors are those organisms which help in transfer of this gene of interest into the host cell. This vector DNA is now sent into our host cell. This is our host cell, its own DNA. This is the host cell DNA, host DNA. So this vector DNA, to send this vector DNA into the host DNA to make it a recombinant cell. Recombinant cells are those cells which contain this recombinant DNA. This vector DNA which have our gene of interest is also known as recombinant DNA. The cells which contain recombinant DNA, they are known as recombinant cells. Now, most of the cell walls, these are the cell walls of host cell. Most of these cell walls, they are impermeable to this foreign DNA because vector DNA is not this host cell's own DNA. So most of the cells, they are impermeable to vector DNA. So what will happen is they do not permit the entry of this vector DNA inside the host cells. So there are many methods to transfer this vector DNA into host cell. This process of transferring the vector DNA into host cell is known as transformation. Transformation is the process of transferring our recombinant DNA inside our host cell. There are various methods of transformation and this gene gun method is also one of those transformation methods. Here what we do is we take our DNA, recombinant DNA, our vector DNA, and we mix it with metal which are in shape of balls, and those metals are either gold or they are tungsten. So very micro-sized balls, they are coated with our negatively charged recombinant DNA, and these metallic balls, they are bombarded onto our target cell. Which is our target cell here? This host cell is our target cell here. So out of like 10 balls, only one ball will get inside this host DNA. So this, whenever this host DNA will have our vector DNA, this cell will become transformed cell because it will have a vector DNA, and this vector DNA will multiply. Then vector DNA will multiply to produce proteins. So the question here says that in biolistic or gene gun method, DNA is coated with what? Metals like gold or tungsten.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.